Again, I wanted to mention my name again. I'm Venkat Chapara. I've been working as a plant pathologist at Langdon Research Extension Center for the past 8 years. Before that, I worked in Minot for 2 years. So I'm pretty comfortable with the northern side of North Dakota's environment. I went, my trials extended from eastern North Dakota to all the way to western North Dakota. So I have a little bit understanding about the weather conditions. And trust me, even though I'll be having 10 years in North Dakota in this position, in the egg experiment stations, each year is different for me, weather-wise, disease-wise, and I felt like I'm in a good area where a lot of crops, most lucrative crop is grown, that is canola, and wherever I go, I'm very proud to say that I'm from canola capital of United States. So we wanted to keep it that way. We will strive to keep it that way. I will do whatever I can. In 2015, when I started in Langdon as a plant pathologist, clubroot was recently introduced to our area. Then we churned our minds with everybody. Then but they said it is a little bit sensitive issue to work on that disease with farmers because people said it's the farmers will take it personally, and we won't get enough cooperation from them. Then trust me, without farmers, I'm nothing over there. I earned their farmers are very welcoming, very nice, and they offered me their land to research that particular disease because clubroot is such a kind of disease. It moves whatever moves the soil. Even my footprints, if something stuck to my pants or foot onto my shoes, I can carry the disease to the new person's field. So that kind of sensitive crop disease I'm working with. But at the same time, I'm also I will be presenting my results, and everybody will be asking, what's the solution? For the most of the deadly diseases which I'm working on, there's not a permanent solution. We have to manage it. We have to go along with that. So I request you guys to to work on that way. We will be very successful in keeping our canola over here. Every help we can get from NDSU or from a special thanks to Northern Canola Growers Association, and all the board members have supported me all through this 8 years. They backed me up to do more research. And they sent everywhere, wherever new information is there for me to learn. So I learned those things. And to start with the clubroot research, the pathogen is so intelligent. It mutates itself, and it is causing a lot of problems to the Canadians. So these board members have sent me to Canada to learn what they are doing. So whatever they are doing, I just borrowed that into our area. Trust me, this year no clubroot fields I have found in our area. So that's the one thing to start with. So today my topic is new sites into canola disease. The prominent diseases I will be talking about in our area. Okay. So I have outlined my topic as a little bit of introduction, which I already did. The lodging, it's commonly, I heard from most of our growers, the crop was very good until the end, and in the end we had problems of lodging. So a lot of farmers here, our good friend, Mr. Joel has invited me to his field. So on, several farmers have invited me to visit their fields across all the northern counties, like 5 to 6 counties. So wherever I went, I saw the same thing. This is the one it caused problems. So we will be talking about. And also my friend Jeremy Saur, he bought the samples way early in July or August. And he said, something is bothering me. So we looked into it. It looked like that particular disease. And we thought, okay, we will work on until the end. So it is that disease. Then the prominent diseases of canola in our area is blackleg and white mold, and we all know what to do with that. The same way we have to work with this new disease which has been recently added to our list is verticillium wilt. And again, we will become, whether it is to call as wilt or stripe, why can't we call it? So I will explain you guys in the later slides. Diagnosis is very important in this case because this kind of disease mimics in one case symptoms. When you look at it, looks like the other disease, and the other things you look like the other disease. So it's very complicated. We have to look into very deep to differentiate which is which. Okay. Then based on that, we have to formulate our management options. Reasons for lodging, in our area, it could be, in general, it could be by several reasons, but whatever it is, lodging causes losses from 16 to 50%. This is a study conducted way long back. The important things which we see lodging because of these reasons: wind, rain, temperature, at flowering. Some varieties has a tendency to lodge, plant population density, nitrogen nutrition, stem diseases, and insect feeding. So there are several things. So that's why I said that we have to figure out what happened. It's because of what factor has influenced this lodging in our area this year. Okay. A combination of genetic, agronomic, and environmental factors cause wide variation in the relative risk of stem and root lodging in canola. So we all have to study all of these things to take into consideration. It won't happen the next year. Okay. Since I'm a plant pathologist, I have to focus more on diseases. If disease is a reason for lodging in canola, what kind of diseases can cause lodging in our area? If lodge due to disease, canola plants are unlikely to straighten up. If it is from something else like environmental factors or varietal character, they might straighten up. But if it is because of disease, they will definitely lodge into the ground. Plants are lodged early due to disease, usually contribute little to yield at harvest. If it is due to disease, it will cause losses in the yield. Okay. And the first disease which we are very familiar with, the yield losses in our area, is blackleg. It causes basal cankers on the stem. You see there, this kind of cankers, and it makes the plants to lodge onto the ground. And we have seen this happens because of internal necrosis in stem and roots that become a lodging factor in our area. It is very prominent. But we have figured out, over the years that there are certain genetics available which we can rotate every year, so we can keep this disease at control. At a certain point, we can manage this disease. That's what we are doing. Our farmers have been doing for so many years. There are new varieties coming up, and the same way the pathogen is also evolution is happening in their genetic structure. They are once we figure out one way, they are developing another resistant character. So on, we are fighting with each other for so long, but still we are able to keep it at certain level. Okay. Then it says one of the statistics if you have 1% increase in blackleg, that might lead to 0.7% of yield reduction. So our first goal in our area to keep this disease at check. Okay, whenever you pick a variety for resistance to which has resistance to blackleg. That is our first criteria. Then the rest of the characters you have to go for. Then there are some areas especially in north and southeastern parts of Cavalier County, we have clubroot over there. So our farmers have to go for clubroot resistance in our area along with blackleg. So that's one recommendation. So at the same time, if you reduce blackleg by 1%, there are chances of increasing of your yield by 5%. So that difference it makes. So you keep that in mind. Blackleg is our first enemy to fight with. Then white mold, everybody knows that there are no genetic resistance available. It has been here for so long. It infects 400 species of broadleaf crops. So the only thing we have is that the pathogen has left us a window to spray from 20 to 30, 40% of flowering. You can go for good chemicals available. And I have a fungicidal data for over the years, which is very effective. If you guys want, I will let you guys later. But as a, there is a good control. The only control we have is fungicides in our area. And I suggest that no matter what, we have to go for one spray of fungicides to manage white mold in our area. Okay. So this is also another statistical Australian research say that for every 1% reduction in plants infected with white mold, there is increased yield by 1.3%. So think about that. But there is always an advantage to go for a fungicidal spray to manage white mold. So what are the diseases we are definitely, have to see that which cause lodging in our area. The first one is blackleg. It's caused by a fungus. The white mold, it's another disease caused by a fungus. And now we can add to our list is verticillium wilt, which it's a very new to our area. In North Dakota, we have found a lot of fields infected with verticillium wilt this year. I was also following the news from Canadian counterparts in Manitoba. They also had the problem. They recognized this problem back in 2014. So, but still, like I thought of adopting whatever they are doing, we would go for those management structures to management options. So, as we got success with implementing their club root management strategy in our North Dakota, the same way we can go for to manage verticillium wilt too. So I'm following them closely. I received a lot of help from them this year in diagnosing the disease. So we are very thankful to have them there. And it's a very actively canola growing country. So they are a little bit ahead of in getting all these diseases than us. So we can follow their management strategies anytime. So this is another picture to show you guys, blackleg, it also causes lodging, white mold, the, white strands here, and they also cause lodging. And see what psyllium stripe, it has broken over here. This is how it caused the lodgings. Mainly I saw this year everywhere. Okay, so this is another confirming factor that we have this disease over here. This is one of the large canola field. I hate to see this kind of fields especially because of diseases. This is the same scenario all over the state wherever I went, and I started scouting for this disease. And a little bit of verticillium stripe or wilt. First of all, scientists were debating that whether we should call this disease as a verticillium wilt or verticillium stripe. So, why? Because usually verticillium pathogen causes in different crops, like take about sunflowers. There is verticillium in that too. But it is species specific, like we call by verticillium dahliae. It's the species which infects sunflowers. It causes wilts in there. Okay, the same genus verticillium, but the species is longisporum. Longisporum means it has long spores. So it is species specific only to brassicas, the canola family. So in here, it doesn't cause a wilt. So we can't call it as verticillium wilt. It will be, oh, where is the wilt over here? Like this year, take it, crop looked very good. It has verticillium infections there. Because it didn't show any wilt to us, we couldn't figure out that we have disease until it lodged. So scientists decided to change its name to verticillium stripe. It's another significant symptom which causes on the canola stems. So they changed the name to verticillium stripe. Since 2016, it's referred as verticillium stripe rather than verticillium wilt. Okay, and it was first identified in 1960. Relative to the other diseases, you can say this, it's a pretty brand new disease compared to other diseases. And found in Europe, Russia, and Canada. In Canada, in Manitoba in 2014. And now in USA. Actually, I have a background story about this. In 2020, we got some farmer calls, and we went to those farmers' fields and collected some stems and brought it to the lab. And I sent it to some different labs, and to my bad, everything came negative to this disease. Then our director, NCGA director, sent me to a verticillium workshop, and I figured out, I have these samples, I have to do differently. So I myself isolated the pathogen in the lab and confirmed with Canadians, and it is positive. So this disease is with us in 2022. So now this year was very congenial for weather we had, and it showed up in a havoc way, very mean way. So, if something comes to our lab, I learned that don't take it lightly. To do as much as you can to confirm that that particular sample has it. Okay. What is this verticillium longisporum? It's a fungus. It's an endophyte. It's generally has been there in the plant system for so long. But what made it as a pathogen? Here, we have intensive farming practices that occur in the absence of crop rotation. This might have triggered verticillium switching from an endophyte lifestyle to a pathogenic one. So too much of exposure to the same kind of environment, same kind of plant, this particular fungus has switched from an endophytic phase to the pathogenic phase. So again, now you can imagine that what kind of management practice you have to practice. All right. So, like as a plant pathologist, I sometimes feel like it's very easy for me to say go for a longer rotation. When I go and interact with the farmers, I know that how hard it is to go for longer crop rotations. Imagine a particular farmer's situation, whether he has an equipment for a new crop to go for or not. So he has been farming in this system for so long. If all of a sudden I say that, go for a longer rotation, it's easier for me to say that, anyhow, if you go, the longer rotation definitely helps. But is there any other things other than that? We have to look for it, right? So this is a relatively new disease. We have to work a long way to come up with some management practice. So until then, you guys should cooperate with me for a longer crop rotation. So it's a high-risk pathogen worldwide. And the typical symptoms are premature senescence and ripening severely reduces yield. Yield reductions reduced between 3 to 34%. Now it's a wide range from 3 to 34%. You can figure out, like how much this year you guys have lost. So it falls into this range or not. And look at these symptoms. Have you ever saw any blackleg infections, when cut across the stem, at the collar region? It looks similar to this. The only difference is blackleg has a very black pith over here, and this is light brownish black. It's not that intense as we see in blackleg. That's the closest we can go for. And that's why I feel like, here it mimics blackleg when you cut it. Okay, even I should confess that whenever I'm evaluating my blackleg trials, if I see this kind of lighter infection, I used to rate it as blackleg, but not as verticillium wilt. So that means I have to backtrack my memory, But yeah, I have been seeing this for quite a while, but I was rating it as a blackleg, and I never educated myself until now. Yeah, so there is a lot to do with this. And here, my friend Jeremy, he got one. Okay. See this symptom. It's a half-leafed, yellowish and half-leaf greenish. Have you guys saw this symptom in the field? Yes. That's how it's during flowering time. You see the flowers? That's where it's the first expression of the symptom. Okay. My friend brought it to the lab and I thought, yeah, this is the one symptom which verticillium might cause. So we have to wait until the end of the season whether we will see any lodging or not. And the next best symptoms, later on, if you see this half yellowing or browning of the stem, half of the stem will be yellow, half of the stem will be greenish. That's another symptom. Okay. And in the end, this is how it lodges. So there are 3 different stages we can go and figure out which kind of symptoms, whether it is verticillium wilt or not. Okay, so if the symptoms are pretty consistent, you can fix. Oh, okay, it is verticillium wilt. And was more symptoms. This is how you see the symptoms here, and at the end of the season, you pick the stem and you see small dots like peppers in there. Those are called microsclerotia. This is the typical character of this pathogen. At the end of its life cycle, it produces those small pinpoint-like structures under the just epidermis of the stem, and that's how they end up either in seed or into the soil, and that's how they overwinter for the next season. And this spore can live up to 10 years. In the same field without a host. Yes, very small. It's not, yeah. Which other pathogen we see the sclerotia? The bigger sclerotia for white mold. So that's why I meant to say that diagnosis is very important. Whether we are seeing blackleg or white mold or verticillium wilt, they all look same at the end of the season. Okay. And this is what, when we split open, these are the black. Still, the picture is not clear, but still we see those. So remember, I'm mentioning about seeing a discoloration in the pith. You have to use this kind of cutter to do that at the color region. If you find this kind of symptoms, yes, that is verticillium wilt. Okay. Yeah, more pictures. See, out of all these things, you can go and easily figure out this is this one we have verticillium on it, and this doesn't have. So you can figure it out. Here, this is another half brownish and half greenish. So at the end of the season, we were able to see those kind of symptoms. When you peel the outer layer, here you see the small pinpoint microsclerotia. This is another thing. And large canola after harvest, this is the most hated symptom by the farmer. Everything large in Rolla County, they mentioned that they have to go opposite way to harvest that and again come back. It took long time for them to harvest than usual. So imagine the gas price and the prices at the time and everything, how much it costed to harvest at that time. So it has a lot of negatives if it is in our field. Okay. Yes, sir. The question is the time of infection, whether the pathogen infects at the same time or as a white mold pathogen or different timing. I'm coming to that, yeah, so. Yeah. So here you see that during the end of the season, I went after swathing or after harvest, I went to the fields to 11 counties around. And I happened to see these kind of stubble. So you see, look at these stubbles. Can you make it out? What pathogen is it? This is white mold. Okay. So here, when you break all that stem all the way to the bottom, here I saw a big sclerotia. This is we know age-old practice. We do that to differentiate white mold, okay, to diagnose white mold. And here, if you look at this, in this row of canola stubbles, I pulled this and look what I saw. This small black sclerotia, it's very fragile. The epidermis is coming easily. Under the epidermis, you see small black dots that is called microsclerotia. That's how you differentiate at the end of the season whether you have white mold on that stem or verticillium. There are a lot of saprophytic forms will be there, and they're exhibiting the same symptoms. But if you can send those samples to me, probably I will try to get some pathogen out of it. So yeah, I can do those in my lab. And this is one nice picture. When I cut across, it is showing a little bit, not complete blackening, light brownish black. This is how verticillium is differentiated. And you see the clean, when I cut it, these are the healthy plants. And this is black leg. See how dark it is. So that's how, another thing you can differentiate between black leg and verticillium. So, yeah. Okay. You see that, as I mentioned, like in 2020 samples, I have received from our colleagues from industry and from the farmers. I sent it to a couple of labs. They came negative for verticillium wilt. And when Barry sent me to that verticillium workshop, I thought, okay, I will work on this. In my lab, I took those samples and put it on the media, like potato dextrose agar, and I got these cultures. Then when I checked the literature, it somewhat looked like verticillium. Oh, I have in my lab these samples, so go on further. So the reliable lab where I sought and I sent my samples is in Manitoba, Dr. Dilharo Fernando. He does a lot of blackleg and verticillium research in his lab. So I contacted him, and his postdoc was kind enough to test our samples. They did the molecular analysis with molecular markers. They did the PCR on it. And out of these 6 cultures, you see that 1A, those 2 are verticillium longisporum. That is our verticillium stripe pathogen, which we got in our samples. So, to convince the scientific world further what we have to do this kind of research too. So I'm not equipped with this research in my lab, so I have to send it to other labs. So it's kind of authentic, what do you call, it's true or not what we did. So this is the molecular research which indicates this. When you have a band over here which matches this kind of bands, this is my 1A. These are the 2 isolates matching verticillium dahliae and verticillium longisporum. So they have Verticillium longisporum in my samples, okay? And lane 1 is the DNA ladder of 100 to 100 base pairs. Lane 2 is my isolate 2A. Lane 3 is isolate 1. Lane 4 is 1A, and the other isolates. And these two are controls. This Verticillium longisporum lane is lane 9 is they have that isolate already identified. So if I get the band same like that, that means we have the Verticillium longisporum. So now it shows the same band as Verticillium dahlia and Verticillium longisporum. So we have to differentiate, right? Right? So they went further. So here they used Verticillium dahlia and Verticillium longisporum differentiation. So Verticillium dahlia is here, the band. Verticillium longisporum is here. So 1 and 1A are the isolates which are exactly matching with Verticillium longisporum. So now it's confirmed that we have Verticillium in our fields. Okay. Again, I would like to thank Dr. Arya Dolatab-Barian from Manitoba. She did all the molecular research, and they didn't charge a single penny to me. So they were very cooperative, and like previously also, they are very good with whatever I send the samples. They do the tests, and they never charge anything for me. Again, to mention, coming to Dan's question, when does the infection occur? So as a typical plant pathologist, we have to study the disease cycle of the pathogen. So where it starts, how it develops, and when it infects, and how it shows the symptoms in the field, and how it overwinters. So we will start with the overwintering structures here, okay? And this is what microsclerotia I was mentioning, the small dots on the stem. They fall onto the ground, and they can live into the soil more than 10 years. And also, they can mix with our seed, infected seed. If you plant the same seed, I know none of our farmers do that, but if you plant the same seed, there is a chances of getting that disease into a new field too. Okay. This is this disease cycle screenshot was developed by Europeans. That's why you will see the seasons: autumn, spring, summer. That's not as fall and summer, winter. Okay. Here in the soil we have this pathogen which overwinters here, and it infects roots at seedling stage. Okay. Just like clubroot, it infects at the very early stage, like small root stage. And then it spreads up the plantum in the xylem during stem extension. As the stem elongates, it moves through the xylem, that's the water conducting channel of the plant. Most symptoms appear as plant starts to ripen. So early ripening, that is premature senescence, okay? That's what we see. And premature ripening of stems, smaller branches, gray discoloration, increases on stem and root. This is the advanced stage symptoms. And once by the end of the season, the stem with infected stem falls onto the ground, and the microsclerotia end up in soil or in the seed. That's how it continues their cycle. So you see this vertical yellow or brown bands on stem. This is the typical symptoms in the later stage of the crop stage. You can find it when the stems are still green. Okay, so if you wanted to know them, look for them. If you are still not sure, I'm at the Langdon experiment station. You can call me anytime. Okay, I can come and check on your fields. So we know everything about the pathogen and how it causes, what are the symptoms, and now the final thing is how do we manage? Again, I mentioned that, longer crop rotation. So if possible, we have to practice it. No effective fungicide treatment is available to verticillium disease. Not only in this crop, so far in any crop like sunflowers or carrots, wherever it causes, we don't have any fungicide to manage that disease. So I hate this part, to say that there is no control. So next is some European research has indicated that there is a Brassica endophyte. It's a bacteria like called Serratia plymoutica F20, strain 20, is effective in managing this disease. So no, I don't have any clue whether it controls or not. I haven't tested, but I read the literature that it says there is some tests have been done on it, and it showed a promising control. Resistance breeding is the preferred strategy for disease management. So since it has been a problem since 2014 in Canada, so there is not much varietal breeding has been done. Probably they are doing it right now. To develop a resistant variety, it takes a minimum 8 to 10 years. Probably they are still in that process. Swathing or straight combining large crop may require some trial and error to see what direction works best. That's what we can suggest now when you are harvesting a large crop. You guys know better than me, how to harvest a large crop. Few verticillium wilt resistance genes have been identified around the world, very few genes, not many. That's why in every crop, or if you could speak about verticillium, there is a minimum control management options in any crop. There are some research scientists have found that there is quantitative resistance. This is a very important one, like with multiple genes that are resistant, had been identified in Brassica C genome of some cabbage lines. Probably, if they can be introduced into our oilseed rape or canola, they might work. Again, I said it's a long shot, like it takes long time, 8 to 10 years. Now, that's all about verticillium or new disease. And this is a club root survey to end with. I have been working on this disease for the past 6 to 7 years. And we wanted to know how far this pathogen has spread from, from Cavalier County or from anywhere where we got the source. So this is a 2021 survey. All the green dots indicates there is no clubroot spores in those soil samples. Yellow dots indicates 1 to 80,000 resting spores. That is the clubroot pathogen spores were there. And red spots indicates greater than 80,000 spores. Okay, if you have greater than 80,000 spores and you plant canola year after year or a 1 year gap and you plant, you will definitely seeing clubroot in 2 to 3 rotations. Like within a 5th year or so, you might see clubroot in your canola if you plant susceptible variety. Okay. Or if your soil has high pH, that is above 7, you may not see it clubroot, even though spores are present in your soil, but you have higher pH, you may not see clubroot in your canola. Okay. Again, along with this resting spore determination, we also did soil pH. What are the pH? So this is a very interesting finding for me, and even the soil scientists liked it. They are saying that North Dakota is moving from basic pH to acidic pH, more acidic pH, and this indicates that all red dots has pH from 4 to 6.6. So, acidic pH is, some no for a lot of crops, I suppose, and they are doing a lot of research on it, the soil scientists. So, my data matches with them too. This is combined. We did this soil survey for 2020 and 2021. Over 500 samples have been collected across the state, and this is how it looks, much more towards acidic pH, and these red dots indicates they have greater than 80,000 spores. Again, I say why they are not saying is probably they have higher pH in those soil samples. And we did, yes. Excuse me? It's a statewide project which was funded by Northern Canola Growers Association. So I had collaborators, myself and in Minot and in Williston, in Fargo. The 5 of us, we collected out of 52 counties in North Dakota, we collected samples from 48. So that's a pretty interesting finding, you know. So it is in soils, but we wanted to know, it's just for a baseline thing. It doesn't mean that you having spores, you will be having infections on your canola. There are a lot of factors should align to see the disease. That's what I, based on my 6 years of research with clubroot, I felt like, oh no, it's not spreading as we expected from northern and southern part of eastern part of Cavalier County to western part of Cavalier County. We haven't seen any in western part of the county. So that's a pretty good sign, you know. We have west high pH towards west, so that's very good, you know. It's contained over there. And some of the neighboring state samples also have it little in mountains. One sample from Minnesota, somewhere close to, it's from vegetable farm. They have some resting spores over there. In Montana, everything is clean. So, and I remember to start with, I said we have 0 clubroot infestations in our county this year, and it's pretty good sign for us. And mainly that's because farmers have opted for an extra crop this year instead of canola. So probably that's the reason we haven't seen any infections of clubroot this year. And we also track different commercially available varieties, whether they are susceptible to clubroot or not. And we also have one mutant pathotype of clubroot pathogen. So if we subject these varieties to that soil pathogen which has mutant pathotype over there, it's good sign that all our resistant varieties handle pretty well. This is for the second year I've tested. So all the resistant varieties of clubroot which are available, they did very good. So these are my controls. They have full disease on them, and these are the ones which I haven't seen any infections on them. These 7 varieties are completely clubroot resistant varieties. And these are the sensitive varieties. 233P as a standard check for me, InVigor 233P. So with this, I wanted to summarize my talk. It's whatever it is, accurate diagnosis of the disease is very important now. So if you guys want guidance, I will be definitely be in my office. You can call me anytime. I will be able to come to your farms and diagnose it. Or if you can send some samples to me, I'll be happy to process them. This year, out of 11 counties, I scouted for clubroot. Along with clubroot, I also scouted for verticillium. Among that, 7 counties had those symptoms of verticillium. DNA analysis has proved that we are positive to verticillium wilt. Verticillium stripe incidences this year in northern ND caused inconvenience while harvest and as well as yield losses in canola. As this is a report from my farmer friends across the state. And clubroot DNA is present in our soils. Again, be mindful. If it is in your soils, it doesn't mean that you will get the disease. If you go on for very frequent canola on canola or canola wheat after canola or something like that, you might be seeing in 5 years or so in your field, the clubroot infections in your canola, if you plant susceptible variety. Rotate canola with other crops 1 in 4 years. So if possible, you can go for it. So that is the so far management option available for many of these diseases. With this, I like to thank all my friends. There are a lot of people, have helped me in conducting my research around. I hope the same kind of support continues further. So I'll be interested in doing more work on these diseases, and hopefully I can find something which is very acceptable for all of our canola community and industries. And with that, I will be happy to take questions. Yep, that is also recommended. So, like Canadians research mostly I'm following, they have recommended whatever they have recommended for clubroot, they are recommending for verticillium. Longer rotation, sanitize your equipment with, all your tires and everything of your equipment should be cleaned, sanitize it, and then go to the next field. So it's a soil-borne pathogen. Yeah, it's very hard. Yeah. That's another good question. We usually don't support to go broadleaf crop after broadleaf every year. Like canola and soybean, we don't recommend. If possible, maintain a small grain in between. That's the reason this year we haven't seen. They had canola in 2020, I suppose. Then 2021, they had wheat. And this year, they have to go for canola in clubroot, in fact, because canola is very good crop for them. They are very good growers of canola. So they wanted, but the weather this year didn't allow them. So they went for either field peas or dry beans, or some even went for soybeans. They all did very good in our area this year. So, that itself indicates like 3-year rotation. Next year, probably they'll go for another wheat, a small grain, then canola. So it's a long interval of 4 years, So if they want, they are going for it. So the weather didn't allow, so it's showing us something, okay, we can contain some diseases this way. No, none. No, probably they are working on it. So some cabbage lines are showing some resistance. So if anything comes up, we will definitely update you guys. Oh, yes. The question is whether we are able to figure out how environmental conditions have played role in getting this epidemic in our area. To be frankly speaking, that's to my surprise also. When we saw in the end that much of infections of verticillium, we have to design a project. It's an epidemiological. It should be an epidemiological project. Our canola pathologist at the campus, he's an epidemiologist. I will try to write a project which aligns, to do some modeling or something like that. So what kind of environmental conditions favors this disease? So looking at this year's conditions, I say that late plantings and frequent rains early in the season has caused sufficient moisture is there. The microsclerotia were in soil, and early stage it's raining, so probably it germinated and it was very ideal conditions. And warm temperatures also in the soil have contributed for the infections. So yeah, probably that's the reason. But it's only my hypothesis. We wanted to pursue that research and see, it's a good question, yeah. We have to work on all those levels. Oh, sorry to answer the, the question is why pH is lowering, right? You mean lower pH is increasing in our state. It's a question for soil scientists. I couldn't answer that. Definitely, I will get back to you by speaking to them. I know good soil scientists in the campus, so I'll see what they will say, and I will give. I have your phone number. I will definitely get back to you. Mm. Oh, okay. Oh, that's good. What probably we should learn from you what you are doing in your soils. Any kind of fertilizer managements or biologicals or anything like that. Yeah. So yes, sir. To manage white mold, you mean? Only for white mold, I recommend to spray at 20% to 30% of flowering of like Proline, Preaxer, even sometimes Quash or and Topsin. They did good in my trials. Yeah. So only for white mold, not for verticillium. Yes, I do. Enduro also good, but yeah, like farmers say that it's a little bit expensive. So I wanted to find a cheaper solutions to them. So I would recommend tops in dry years, like last year, not in 2022, 2021. Several farmers were asking me what to spray, and I also follow the NDSU forecasting model. Based on that, I recommended some farmers very slim chances of getting white mold this year. Go for Topsin or Quash. Topsin worked good. Oh, that's another good question. It's depends upon the equipment you have, right? I read some papers like even if it's spread in either all the directions, there are some equipment is available which can pick up everything and it can combine pretty good as a swathing too. There are not big differences between swathing and straight cut.